So for those who aren't familiar, zoonotic diseases are diseases that are transmitted from humans or from animals into humans. They're attributed to about 60% of known human infectious diseases worldwide, as well as 75% of emerging infectious diseases. Most zoonoses are maintained in wildlife and the form of reservoirs, and many zoonotic pathogens are also vector-borne, especially tick-borne. Tick-borne diseases now account for more than 75% of all vector-borne pathogen cases in the U.S. And many vector species, including rhizoid scapularis, as you can see in this graph, are expanding in both range and abundance. This expansion is driven mostly by climate change, land use, and increasing wildlife populations. And despite this, there's still limited public knowledge about the tick-borne pathogen disease risk in places like the Midwest. Reservoir hosts that maintain pathogens act as a source of the infection, playing a central role in disease systems. These infections typically cause little to no pathology in the raccoons themselves or in reservoirs themselves, allowing them to maintain transmission over a long period of time. Without infected reservoir hosts, we wouldn't have infected ticks. So they really do play a critical role in the life cycle of vector-borne pathogens. Because of this, understanding infection dynamics in reservoir hosts is essential for understanding. Just kind of showing the example of like kind of what reservoir hosts are and how they play a role in the life cycle of tick-borne pathogens. To understand disease risk, we need to understand what's driving pathogen prevalence. And there are many factors that can play a role in shaping disease dynamics. One of them includes host population size, which is something that I focused on in my study. There are two measures of host population prevalence that can have varying effects on pathogen prevalence. And those include host density, which is the number of individuals within a specific unit area. And then we have host abundance, which is the total number of individuals within a population. High host density can lead to increased animal contact rates, and that can facilitate pathogen transmission, which is especially important for pathogens that require direct contact for transmission. Alternatively, high host abundance can facilitate parasite persistence in the environment and also facilitate spread to new locations as more animals are around in the habitat. Urbanization is another major driver of infection dynamics. As land development rapidly increases with the increasing global population, we see changes in host dispersal patterns, altering species composition, and altering contact rates of hosts and their pathogens. And this can all play a role, obviously, in disease transmission. There's also been an increase in densities of many synanthropic species, so such as raccoons or skunks, and these can increase human-wildlife interactions, which of course plays a role in that zoonotic disease risk. However, the effects of urbanization can be complex and not always consistent, so this warrants further investigation. As I mentioned, one important synanthropic species is raccoons. So raccoons are mesopredators that are distributed widely across the United States. They thrive in urban settings due to the abundance of anthropogenic resources, placing them in close proximity to humans. Their reliance on anthropogenic resources in urban areas, again, places them in close proximity to humans, making them especially relevant for public health. Some of these zoonotic pathogens that are known to be carried by raccoons are tick-borne pathogens. In the state of Illinois, there are 3 main tick vectors that transmit pathogens, including the Lone Star tick, the American dog tick, and the black-legged or tick tick. So these tick vectors carry things like Ehrlichia japonicus, which is the causal agent of ehrlichiosis, Babesia macroti, which causes babesiosis, Anaplasma phagocytophilum, which causes anaplasmosis, Borrelia burgdorferi, which causes Lyme disease, which I'm sure most people are probably familiar with, and finally, Rickettsia or Rickettsia, which causes rock. Another zoonotic pathogen found in raccoons is raccoon round. This is an intestinal nematode found in raccoons, and it's widely distributed across the U.S. Transmission to adult raccoons occurs through ingesting third-stage larvae, that are in the tissues of peritoneum, such as like white-footed mice. And then juvenile raccoons can become infected by ingesting impregnated eggs that are passed in feces. This parasite is particularly important because of its ability to infect humans. It can cause severe neurological disease, which can be deadly. And there have also been cases documented in 11 states, including Illinois. Human transmission occurs through the ingestion of impregnated eggs, which places children at highest risk due to their poor hygiene habits. I'm sure you're all aware they like to put their hands in their mouths when they're outside in the soil. So most of these cases have been in children. So some raccoon behaviors are especially important that contribute to transmission risk. So raccoons use communal latrines, and this can lead to heavy environmental contamination when repeatedly being used by an infected raccoon. Raccoons also typically have smaller home ranges in urban areas when compared to rural areas, and this can actually limit the dispersal of the pathogen and kind of keep it in its contained space. And then again, raccoons heavily rely on genetic sources when they're available. So this is especially important to the raccoon roundworm transmission. As I mentioned, adult raccoons can be infected by ingesting an infected peritoneum host. So if they're relying on human food sources rather than predation, then that can disrupt that transmission. Monitoring pathogens in reservoir hosts is critical for public health. Human cases of tick-borne diseases have been tripled since 2004, and cases of raccoon roundworm are also increasing. At the same time, distributions are also expanding into new regions. So understanding pathogen prevalence in wildlife and reservoirs is essential for predicting and mitigating risks. Despite this, there are important gaps in our understanding. Disease monitoring of tick-borne pathogens in raccoons has been mostly confined to the southeastern U.S. due to the previous tick distribution. There's also limited research examining the role of urbanization and host population size on tick-borne pathogen prevalence in raccoons, and studies examining the same relationships with raccoon roundworm have produced conflicting results. And so my research is aimed to address these knowledge gaps through three main aims. So this brings us into the first aim, which is to evaluate the effects of urbanization and raccoon population size on raccoon roundworm. My objectives were to quantify the prevalence of raccoon roundworm in Northern Illinois raccoons, assess the relationship between raccoon roundworm and urbanization, and then finally assess the relationship between prevalence and raccoon populations. Sample of raccoons across 5 sites representing a rural to urban gradient in Northern Illinois. Just to orient everyone, the star on the map is Chicago. If you're not super familiar with Northern Illinois, so my sites included the Chusa Grasslands and Richardson Wildlife Foundation. These sites are mainly surrounded by agricultural fields, and they also have no public access, reduces the human footprint at these sites. And then my suburban sites included Hickory Grove Forest Preserve, Pine Dunes Forest Preserve, and Spring Bluff Forest Preserve. These sites are all mainly surrounded by residential and commercial lands, and they're also public parks. So a lot of these have picnic areas dispersed throughout hiking trails. So of course, providing more access to allergenic food sources. Between April and June of 2022 to 2024, raccoons were captured and removed by our site collaborators. I collected a total of 280 raccoons across all 5 sites, and I performed necropsies and I examined the intestinal tracts for parasites. For my analysis, I first calculated prevalence as the proportion of infected raccoons from each site. And then for my raccoon abundance covariate, I calculated catch per unit effort, or CPUE, as the measure, or I calculated as the number of raccoons captured per 100 nights of capture effort at each site. I then calculated raccoon density using the less depletion model. which basically just estimates the size of an exploited population within an effective trapping site area at each site. And then for my urbanization covariate, I calculated the proportion of developed land cover. And I did this within a 0.8 kilometer buffer, and that's supposed to represent a raccoon home range in urban areas. And I did this around each site using the National Land Cover Database, and I included all developed land cover classifications. And then to investigate the relationship between urbanization, raccoon population size, and raccoon roundworm prevalence, I used a generalized linear model and included CPUE, raccoon density, and percentage developed land as estimates. So out of the 280 raccoons that are collected across my 5 sites, I detected raccoon roundworm in 106 individuals. So that's an overall prevalence of 38%. And if you're looking at this graph, the green sites, the green bars represent my rural sites and the blue represent my suburban sites. And as you can see, raccoon roundworm prevalence varied from about 25% to 49% across my sites. So I did not find a significant relationship between raccoon density and percentage developed land. But I did find that lower raccoon roundworm prevalence was associated with higher raccoon abundance. One explanation for this inverse relationship could be parasite aggregation, and this is the tendency for parasites to aggregate in a smaller number of individuals as the host population size increases. I compared my results to a similar study out of the Chicago metropolitan area. So in this study, they had 3 study sites that they categorized as rural, suburban, or urban, and they used these categories for their analysis. Their rural site is actually more similar to my most urban site because 1, it's proximity to Chicago, and 2, it's also a public park with lots of picnic areas and stuff like that. Most of the studies that have looked at this relationship and used categories of urbanization have not included the criterion that they use to create these classifications. And so because of that, out of 3 total studies, they've all found different relationships between raccoon roundworm and urbanization. However, between these 2 studies, mine and Page study, they both found that high raccoon abundance was associated with low raccoon abundance. So in summary, high raccoon abundance was associated with low raccoon roundworm prevalence. This suggests that reducing the raccoon population size would be ineffective to reduce human infection risk. And also the lack of a clear trend in the relationship between urbanization and raccoon roundworm prevalence highlights the need for standardized land use classifications and definitions of urban, suburban, and rural environments across them so that we can better compare across studies. And then also further analyses examining this relationship with finer scale descriptors of landscape are really needed to understand its driving prevalence and raccoon. An example of this could be anthropogenic resource availability. I always make a joke that I wish there was a trash layer on GIS that we could put here because I feel like that might show us more about its driving prevalence since it really comes down to raccoon. So this brings me to my second research aim, which was to assess patterns of zoonotic pathogen prevalence across a rural to urban gradient. So this is essentially the same thing I did in my first chapter, except I focused on 2. My objectives of this chapter were to quantify the prevalence of zoonotic pathogens in northern Illinois raccoons. And the pathogens that I focused on were Babesia macroti, Anaplasma phagocytophilum, Ehrlichia chapayensis, Rickettsia, and Borrelia. I then assessed the relationship between pathogen prevalence and urbanization, and then assessed the relationship between prevalence and raccoons. Again, I collected these 280 raccoons, and I collected samples of the heart, liver, and spleen. I then extracted DNA from these tissues and analyzed for pathogen prevalence using qPCR and pathogen-specific primers and probes. Prevalence was again calculated as the proportion of infected individuals from each site. And then for pathogens that have moderate prevalence, I again used the generalized linear model and included CPUE, percentage developed land, and raccoon density as fixed effects to examine these relationships. But for pathogens that had skewed prevalence, I investigated these relationships using Brooks penalized logistic regression to reduce bias and maximum likelihood estimates in the presence of rare outcomes from separation. So I did not detect Ehrlichia chapiensis in any of my raccoons across my 5 sites. And I detected low prevalence of both Borrelia burgdorferi and Rickettsia rickettsii. For Borrelia burgdorferi, I only had 2 positive detections, both of which came out of my site that had the least amount of land development. And then for Rickettsia rickettsii, I only had 1 positive detection, and it was again in one of my multiple sites. However, I did see moderate prevalence of anaplasma phagocytophilum, and this varied from 23% all the way up to 55% across sites. And then I found extremely high prevalence of Babesia microti-like across all 5 sites, with 3 of my sites being 100% uninfected. When comparing the pathogen prevalence observed in my study to previous studies of raccoons, I found similar prevalences of Ehrlichia chapiensis and anaplasma phagocytophilum, similar to what's been documented in the Southeast. The prevalence that I observed of Rickettsia rickettsii and Borrelia burgdorferi, however, were lower than what has been observed in New York. And then finally, the prevalence of Babesia microti-like that I observed in my study was higher than what's been observed across the United States and Canada, and it was more consistent with the prevalence estimates out of North Carolina in the same study. Raccoon population size didn't have an effect on tick-borne pathogen prevalence in both raccoon density or raccoon abundance. However, I did find that a higher percentage of developed land was associated with lower anaplasma phagocytophilum. We found the same relationship between land cover and Borrelia burgdorferi pathogen prevalence. But I wanted to note that this was driven by 2 positive detections both at the same site. So this likely contributed to an inflated effect size estimate, even with the bias correction. Because of this, we would need further sampling to confirm this. So the high prevalence of Babesia microti-like across northern Illinois suggests a high level of illness. This high prevalence that I observed in my raccoons could be explained by a southward shift in infected raccoons from Wisconsin. As you can see in this map, it's actually considered endemic in Wisconsin. And this would be consistent with the southward shift from northern Wisconsin into southern Wisconsin, where again, this pathogen is now considered endemic. And you remember, all of my study sites were in pretty close proximity to Wisconsin. One of them actually extends into the southern part of the state, just a tiny little bit. And so this suggests that these infected ticks are moving south, and Illinois could be the next endemic area. Also, the prevalence of anaplasma phagocytophilum found across northern Illinois, coupled with the uprising cases that are coming specifically out of northern Illinois, suggests an increasing public health risk. But my findings also did suggest that there's lower risk of infection in urban areas. This lack of a consistent relationship between pathogen prevalence and urbanization in the study shows that prevalence could be driven more by host community composition than by the size of a single host species. So most host communities are made up of reservoir hosts of different competencies. So we can have incompetent hosts that are not as effective at transmitting these pathogens into the ticks. And then you can have more competent hosts that are really effective at transmitting these pathogens into ticks. So if we have higher prevalent or higher proportion of competent reservoirs than we do incompetent reservoirs, this could amplify the pathogen prevalence in the host community. Whereas if we have a higher proportion of incompetent hosts, this could dilute the pathogen. So these are things that should be looked at when trying to assess drivers of prevalence in specific communities. So finally, my final research aim was to assess co-infection structure and pathogen richness across a rural to urban gradient So just kind of combining everything to some. So hosts are rarely infected with a single pathogen, and instead often harbor multiple pathogens simultaneously. Co-infection patterns can result from both synergistic or antagonistic interactions between subpathogens. So infection, in some cases, infection with one pathogen can increase susceptibility of a host to a secondary infection due to altering their immune function. And then in other cases, pathogens can compete with each other, making it less likely for both of them to infect one individual. Patterns of co-infection can also arise due to shared ecological drivers, so like habitat features, vector communities, and also host behaviors. And however, the role of urbanization shaping these patterns of co-infection remains poorly understood. Borrelia burgdorferi, Babesia microti, and Plasmodium cytophilum all share the same tick vector, which is Ixodes scapularis, and this could promote co-infection in both ticks and in reservoirs. Positive associations have also been documented between Babesia microti, Borrelia burgdorferi, and Plasmodium cytophilum in Ixodes ticks, and increased transmission of Babesia microti to ticks feeding on hosts that are co-infected with Babesia microti and Borrelia burgdorferi have been observed as well. However, patterns of co-infection in reservoir hosts is less understood, with results of different studies demonstrating independent transmission as well as positive and negative interactions between Babesia microti, Borrelia burgdorferi, and Plasmodium cytophilum in small animals. So the objectives of this chapter were to examine pairwise co-infection patterns among pathogens to test whether infections co-occurred more or less often than expected after accounting for environmental factors. And the second objective was to quantify pathogen richness to assess whether raccoons in more urbanized areas or at higher abundances carried greater or fewer pathogens per individual. So first, I examined whether pathogens tended to co-occur within raccoons more or less often than expected after accounting for environmental host factors using a hierarchical multivariate probit regression, which modeled binary infection outcomes simultaneously and estimated residual pathogen associations after controlling for these environmental host covariates. As a complementary analysis, I used a permutation-based null model to test whether observed co-infection frequencies deviated from random expectations while also preserving cycle requirements. So next, I examined whether raccoons in more urbanized landscapes or at higher abundances carried more or fewer pathogens per individual. So first, I calculated pathogen richness as the number of pathogens per raccoon. I then modeled pathogen richness per raccoon as an ordered categorical response using an ordinal regression model, and predicts probabilities of richness categories were estimated across urbanization and CPU gradients using bootstrap 95% confidence. So this heat map here is just showing you the prevalence of pairwise co-infections detected in raccoons across my study sites. And overall, I found 166 pathogen co-infections, and this accounts for about 60% of all of my infected raccoons, which was 279 out of 280. I also observed 38 triple infections between raccoon roundworm, Anaplasma cytophilum, and Babesia microti-like. And I also observed one quadruple infection between raccoon roundworm, Borrelia burgdorferi, Anaplasma cytophilum, and Babesia. So while co-infections were common, I did not detect any strong pathogen associations after accounting for environmental and host covariates, as you can see in this figure, showing the residual pathogen associations. This result was also supported by the permutation-based null model, which showed the co-infection frequencies did not deviate from random expectations. These results suggest that co-infection is not driven by strong interactions among pathogens within the host, but instead likely driven by shared exposure. In terms of pathogen richness, I did not find that raccoon abundance had a significant effect on pathogen richness. However, I did find that raccoons in more developed areas were more likely to carry fewer pathogens than raccoons in more or less developed areas. These results suggest that urbanization could be acting as a filter of pathogen communities, reducing pathogen diversity, likely through changes in host communities and also in vector habitat. So the high co-infection rates that I saw indicate raccoons are frequently exposed to multiple pathogens. However, host abundance alone does not seem to structure pathogen communities. And this kind of goes back to what I was saying earlier about having that larger host community that has varying levels of reservoir competency within this host community. So that's likely what's structuring these pathogen communities more than just the abundance of one single host species within the community. Co-infection is likely driven by shared exposure rather than strong pathogen interactions. And then again, urbanization appears to act as a filter. And this is likely driven by reduced vector habitat and altered host communities, especially in urban areas. Lower pathogen richness in urban areas may reduce multi-pathogen exposure risk, but it's also worth noting that highly prevalent pathogens, like what we saw with the Prevalence microti, did persist even in developed areas. So overall, raccoons across northern Illinois harbored a very high prevalence of zoonotic pathogens, with nearly every raccoon infected with at least one pathogen. I actually only had one raccoon in my study that didn't test positive for anything. The rest of them were infected with these pathogens. This high raccoon abundance was also associated with low raccoon roundworm prevalence, and as I mentioned, with the high prevalence of Babesia microti-like, it was detected in nearly all individuals. Co-infections were also common, and this happened in about 60% of high infected raccoons. And then I found that urbanization reduced pathogen richness but didn't eliminate risk. So all of my results point to pathogens really being shaped by interactions among multiple host species with varying abundances, reservoir competencies, and ecological roles. And then co-infections in these reservoir hosts are likely driven by shared exposure rather than pathogen interactions. So these results suggest that raccoons can serve as sentinel hosts for zoonotic pathogen surveillance. Nuisance animals like raccoons are frequently removed from wildlife or through wildlife management and control programs, offering up a cost-effective and logistically feasible opportunity for ongoing surveillance. And as we've seen in my study, because of the fact that they were commonly infected, they could provide information about shifts in pathogen prevalence before we really see that increase in human health risk. The reduction of raccoon population is probably an ineffective tool to reduce human infection risk of both tick-borne pathogens as well as raccoon roundworm. And so instead, management could focus more on education. So national education campaigns, increasing public awareness to tick-borne pathogens, raccoon roundworm, and first prevention methods could be an effective way to mitigate human health risks. And then also, both people and agencies could take measures to limit raccoon access to human-provided resources, and that can keep raccoons out of human spaces and reduce that interaction among humans and raccoons. And then finally, there's a need for targeted latrine removal and decontamination to remove environmental contamination of raccoon roundworm, and this would significantly decrease public health risk and also decrease the prevalence of raccoon roundworm, especially in juvenile raccoons. So first, I'd like to acknowledge my advisors, Dr. Jimenez and Dr. Nielsen, as well as my committee for support over the last 5 years. And then my undergraduate students and field technicians that helped me out in the lab and in the field collecting data. My fellow grad students for emotional support. And then also I would like to thank my funding sources with the Illinois Department of Natural Resources, infringing intrusive grasslands, as well as all of our collaborators across sites. And then, of course, I would like to thank my boyfriend, my family, my friends, who like to dress up as Shrek, go out to bars, and of course, my adorable dog Finn for all of the support over the last 5 years. Take any questions.